Swayam Prabha. Digital India. Educated India.
Hello and welcome to our today's lecture of Introduction to Mechanobiology. So, in the last few lectures we had discussed about different modes of migration, specifically focusing on three different aspects, some example of mesenchymal migration and how actin dynamics drives mesenchymal migration, one study on amoeboidal migration and another one on collective cell migration in the last lecture, where I had focused on leader and follower cells and also this phenomena called coherent angular rotation when you confine epithelial cells within patterned regions. Okay. So, as part of the reading assignment, I would like to refer you to these two papers. Also, the PNAS paper which we discussed about the using stem cells for studying wound healing, and also this second paper where I just touched upon about coherent angular rotation, and I request you to go through this paper in great detail. Okay. Having said that, I would now switch gears and stop our discussion from migration to some other aspects of cell behavior specifically. So, I will start with behavior of stem cells or mechanobiology of stem cells and go on to mechanobiology of diseases over the next 2 weeks. Okay. So, having said that, so today we will start discussing about mechanobiology of stem cells. Okay. We want to talk about stem cells. Okay. So, what are stem cells? So, stem cells are undifferentiated biological cells. Okay. You have cells which can differentiate. Okay. So, two aspects of cells, they can self renew, okay, or divide to generate more of their own type, or you can differentiate into specialized cells of different tissues. Okay. So, in case of mammals, you broadly have two types of stem cells. Okay. Embryonic stem cells and adult stem cells. Okay. So, what are ESCs? Okay. ESCs are cells which are isolated from the blastocysts. Okay. So, these embryonic stem cells, so they are isolated from blastocysts, and these guys are called pluripotent because they can generate cells of the ectoderm, okay, mesoderm and endoderm. Okay. So, embryonic stem cells can generate all cells of all different germ layers, but there is some ethical issue about embryonic stem cells. So, what is the ethical issue? You have these cells which are isolated from an embryo. So, by isolating cells from the blastocyst stage, you are actually killing that organism. Okay. So, that is the ethical issue. That is the main primary problem why embryonic stem cells did not find much use for regenerative medicine applications. Okay. In contrast, you have adult stem cells. So, these stem cells reside at multiple locations. Okay. And so, you have isolation of adult stem cells. One of them is from bone marrow, one of them from adipose tissue, okay, you can isolate them from blood, okay, or you can isolate from umbilical cord. So, this is right after birth. Okay. So, the advantage of adult stem cells is you do not have these ethical hurdles, okay, and you can in principle. So, because these cells are autologous, so you can obtain the cells from your own body and use it either for banking, so that you can use it for future surgical procedures, okay, as well as to re-engineer them, to differentiate them into given cell types and then re-inject them back into your body. So, you will not, these cells will not elicit an immune response because they are from your own body. So, that is the promise of regenerative medicine, you can use it. So, if you compare the ES cells with adult stem cells, so you can have different, so ES cells, ESCs are called pluripotent. They are called pluripotent because they have the potential to differentiate into any 3 cell layers. Okay. They have the capacity to differentiate into cells of 3 germ layers. Okay. So, which are endoderm, okay, that means stomach, okay, lungs, etc. Mesoderm, which is muscle, bone, etc., or ectoderm, which include epidermal and nerve tissues. Okay. So, ESCs are pluripotent. In comparison, you have adult stem cells, they are what is called as multipotent. Okay. So, their ability to differentiate into all different cell types is not possible. Okay. So, for example, if you have a hematopoietic stem cell, H is for hematopoietic stem cell, so it can give rise to all blood cell types including lymphocyte, okay, monocyte and neutrophil. Okay. But this guy, it cannot give rise to a bone cell. Okay. So, this is called a multipotent. Okay. And in comparison to pluripotent or multipotent, you have one more case which is called totipotent, and example is the zygote. Okay. So, these can also give rise to your sperm cells. Okay. So, this is totipotency. In addition to, so the way the stem cell hierarchy was initially imagined, people would always thought that from this stem cell you gave rise okay, you have these cycles. So, this guy can self renew or differentiate. Okay, suggesting that you can only traverse the tree in this direction or downward direction. But this was changed by the seminal work of Yamanaka, okay, Shinya Yamanaka, very recently. So, he showed that you can generate induced pluripotent stem cells. Okay. In short, they are referred to as iPS cells. Okay. So, what iPS cells are? They are reprogrammed. So, you can take a terminally differentiated cell like a fibroblast and you can reprogram it to generate the original stem cell. Okay. So, these are reprogrammed, where the mature cells are reprogrammed to become pluripotent. Okay. So, what are the strengths of this IPS technology? Because you can in principle iPSCs you can derive from adult tissues. So, not only does it bypass the need for embryos, but you can be made, these can be made in a patient matched manner. Okay. So, in what does this mean? That each patient can generate his her own pluripotent stem cell line, okay, and this is what is the beauty of personalized medicine. Okay. The other interesting aspect of iPSCs is you can simulate diseases which take long term to manifest and cannot be studied in vitro using iPSCs. Okay. But still they are far from that stage. You can think of therapeutically engineering iPS cells for personalized medicine. So, this is just a broad introduction to stem cells. Now, typically if you look at all these cells, okay, these different cell types, a muscle cell is more elongated, okay, a fat cell. So, this is the shape of a fat cell, a muscle cell, a nerve cell, okay, versus, okay, a bone cell. Okay, so you have the nuclei at the center. Okay. So, what you see that these cells have distinct phenotype. Okay, they are phenotypically distinct. Okay. So, though they descend from a common MSC, you have an MSC from which they originate. So, mesenchymal stem cell precursor is the same, okay. You have distinct phenotypes for each of these cells. So, in other words, shape seems to be correlated to cell type, and each of these cell types have a given function. Okay. So, the question here then becomes, okay, that is it possible that function of cells is directly related to the shape, okay, and what is the nature of the dependency? Is it that shape determines function or function determines shape? Okay. So, that is the question to be asked. Okay. So, typically we always think that genotype dictates phenotype, but however, is it possible that by changing the phenotype you can alter the function of stem cells? Okay. We will discuss one such paper, okay, in the context of HMSC. So, HMSC differentiation, okay, human mesenchymal stem cell, okay. So, these guys are known to differentiate into adipose or fat cells as well as osteoblasts or bone cells. Okay. So, now one of the striking observations in these experiments is that the decision to differentiate into an adipose cell versus an osteoblast is often dependent on the cell density. Okay. The density at which these cells are plated seems to have a strong correlation in the lineage to which these cells differentiate. Okay. So, of course, one would imagine that growth factors are the only things which determine the overall response. Okay. But cell density, what is the correlation between cell density and growth factors in relation to this differentiation? Okay. Is it possible? So, imagine this picture, okay, that cell density actually impacts the cell shape and cell shape then impacts the differentiation status. Okay. So, we are saying that it is the phenotype. So, you are kind of indirectly controlling phenotype by cell density, and then this phenotype is what dictates this overall response. Okay. So, what the authors started off was they did these experiments in which you cultured cells, okay, HMSCs, were plated at different densities. Okay. Either at 1000 cells per centimeter square or 25 times higher, 25,000 cells per centimeter square. Okay. And then they also added one more factor, which is your soluble Q or growth factor. They also did these experiments in the context of adipogenic media, okay, which would typically induce differentiation of HMSCs into adipocytes, or osteogenic media, okay, where the chemical factors present in this media induced osteogenic differentiation. Okay. Now, what they found was when they plated cells at this low density, all the cells HMSCs differentiated into osteoblasts. Okay. And even so, addition of adipogenic media did not have any influence. In contrast, at the cell seeding density of 25,000 per centimeter square, you had differentiation into adipocytes. Okay. So, what you find is, as a function of time, at low density you get osteoblasts. So, low density leads to osteoblasts, high density leads to adipocytes. So, suggesting that, again demonstrating that cell plating density influences MSC fate. Now, if we are to look at our argument of cell density indirectly impacting the cell shape. So, one of the things what you are doing is, in a given area, let us say in this given box, I plate a single cell versus, so this is one cell plated in this box versus 10 cells plated in this box simultaneously. Okay. So, what you see that one of the indirect consequences of plating cells at a high density would mean that the allowable area for a single cell to spread is lot less. Okay. So, and also, so first of all, your cell spreading area is low here, and here cell spreading area is high. Okay. Also, you have this phenomena called contact inhibition. Okay. And in this phenomena, you have cell division ceases when cells contact each other. Okay. So, in a sense, what you see here, what this shows you is that density indirectly influences cell shape. So, is it possible that it is this cell shape or cell size which determines the nature of the differentiation trajectory? So, however, this particular experiment, it is difficult to answer that question, because it can very well be possible that when you have high density, the growth factors, other things that the cells secrete might have a role to play. Also, the cells touching each other might also have a role to play in this adipogenic differentiation. So, to ask if cell shape alone, so the question is, can cell shape influence MSC fate? Is the question. So, to ask that question, what the authors did was they again using microfabrication. So, they used microfabrication, and what you have in microfabrication, in a background of surface, you can pattern. So, you have seen what is microfabrication earlier in the context of focal additions. You can pattern these islands either with very small dots, or you can have, let us say, you can make these square patterns with bigger area. So, these areas are adhesive, and so this is adhesive, and this area is non-adhesive. So, what they changed was the area of the island. So, island area was, island size and area were changed. So, what they found was, when they varied the island size, this is island size, and let us say it is the y axis is the extent of differentiation. So, what you see, what the authors found was, with increase in island size, they observed the following thing. This is osteogenic and this is adipogenic. Increase in island size switched the differentiation status with a more prominent adipogenic differentiation on small island sizes. So, this environment will be more adipogenic and this environment will be more osteogenic. So, bigger the island size, more osteogenic differentiation, less adipogenic differentiation. Smaller the island size, more adipogenic differentiation, less osteogenic differentiation. Now, when they looked at the cells, so if they look at the cells, you had a strong stress fibers in case of cells which differentiated into osteogenic lineage, and in case of adipogenic in small islands, so there was no stress fibers. So, again going back to the analogy of cell as a tent, so greater amount of stress fibers. So, if I were to draw tension, the level of cytoskeletal tension, this is low, this is high. So, again you have this correlation between tension and the lineage differentiation. So, in adipogenic, so in low, when tension is low, you have adipogenic differentiation. When tension is high, you have osteogenic differentiation. So, they then asked that, can differentiation status be regulated by perturbing tension. Okay, is the question that they asked. And what they found was, again, so they perturb tension using, so you have rhokinase, okay, is one of the signaling pathways which directly mediates cytoskeletal tension. Okay. So, what they found, okay, compared to control, if they added, okay, two drugs, Cyto D or Y27632, okay, Y27632 is a ROCK inhibitor and Cyto D is an actin depolymerizing agent. Okay. Once again, adipogenic differentiation dropped. Okay. So, this is my adipo differentiation. Okay. So, you lower the tension. Okay. Sorry, this is, yeah, so this is my, no, so this is osteo differentiation and this is adipo differentiation. Okay. Lower the tension. So, if you lower the tension, your osteogenic differentiation drops. If you lower the tension, then your adipogenic differentiation increases. Okay. So, these results show that MSC fate depends on cytoskeletal tension. Okay. So, with that I stop here. So, to summarize what we discussed today, we showed that in case of stem cells, in mesenchymal stem cells, by perturbing things like shape or tension, you can perturb its differentiation pathways, okay, with more amount of tension leading to an osteogenic differentiation and lowering of tension leading to adipogenic differentiation. And this changing of tension is also associated with changing of the cell shape. So, in case of low tension, the cells are more rounded, do not have much cytoskeleton, versus when they are highly tensed, you have structures which are more elongated, more stretched out configurations of the cell. Thank you for your attention.